Ready!
Hey everyone, we are the Study Buddies. Welcome back to our channel. In today's video or biology edition, we will be learning about another new subtopic, which is Eukaryotic Cell Part 1. In our previous video, we have learned about prokaryotic cells and also about endosymbiotic theory that led to the formation of another type of cell, which is eukaryotic cells, or the cells with membrane-bound nucleus and organelles. The main difference between prokaryotic and eukaryotic cells is the idea of compartmentalization. So what does this idea of compartmentalization actually all about? Let me give you an example. Imagine prokaryotic cells as a house with no rooms at all. It only has one big space where all the cell functions take place. Eukaryotic cells, on the other hand, is like a house with many rooms in it. Each room has its own function to perform. The rooms we are referring to are the organelles in the eukaryotic cells. These rooms or compartments in the eukaryotic cells are essential as they aid in complementing with the complexity of the cell's function. Compartmentalization in eukaryotic cells is largely about efficiency. Separating the cell into different parts allows for the creation of specific microenvironments within a cell. That way, each organelle can have all the advantages it needs to perform to the best of its ability. So, with having the idea of compartmentalization as the preface, let's look at the generalized depictions of an animal and plant cells. Fundamentally, animal and plant cells have several similar structures in them, such as the nucleus, plasma membrane, mitochondria, ribosomes, rough and smooth endoplasmic reticulum, which I will refer to as RER and SER further in this video, Golgi apparatus, which I will refer to as GA, and many other structures. Some of the structures that differ in them would be: only animal cells have centrioles and lysosomes, while only plant cells have cell wall, chloroplast, and a central vacuole. Their differences are suitable with their respective needs as well. Another important concept we are going to learn about is Organelle density. Cells can vary in proportion of organelles they have depending on the specialized function of the cell. For example, liver cell, whose function is partly to detoxify drugs and other ingested compounds, contains a greater proportion of smooth endoplasmic reticulum or SER. This is because one of the functions of smooth endoplasmic reticulum is to aid in detoxification of drugs as well. Another example is a sperm cell who needs to swim through a longer distance to the uterus, has a greater number of mitochondria that generates energy for its propulsion. The key thing or tips to remember here is that always connect the need of the cell being highlighted to the function of the organelle. Now, let's try answering this question quickly. What organelle do you think is most abundant in pancreatic cells? If your answer is rough endoplasmic reticulum or RER, then you're right. This is because the main function of pancreatic cell is to produce pancreatic juices filled with various enzymes. So, the organelles that most probably responsible in producing enzymes as a final product would be the RER. So, that's how you connect the function of the cell to the organelle. Now, let's look at the first structure in the eukaryotic cell, which is the nucleus. Most of us may have learned before that nucleus is the control center of a cell. To make that statement even accurate, nucleus is actually the place where the genetic information or DNA is stored in the form of a chromosome. A nucleus has a nuclear envelope that separates its environment, while substances can still enter and exit the nucleus via the nuclear pores. The place stained darker here is called the nucleolus. It is the site where ribosomal RNA is produced to form ribosomes later in the process. The next structure we have is called the ribosomes. ribosomes are the site of protein synthesis. Ribosomes can be found freely scattered around the cell or attached to the RER where proteins synthesized in the ribosomes are transported. A ribosome is made up of a large and small subunits. These two subunits run over a strand of mRNA from the nucleus and assemble amino acid chains to be made into actual protein later. Nextly, we have a very important system called the endomembrane system. It consists of the nuclear envelope, membranes of endoplasmic reticulum, the Golgi apparatus, and vesicles. This system compartmentalizes the cell so that particular enzymatic reactions are restricted to specific regions and overall cell efficiency is increased. Firstly, we have the endoplasmic reticulum, or let me refer it as the ER. It is actually a continuous channel physically continuing with the nuclear envelope. So, we have two types of this ER, which is the rough ER and smooth ER. The rough ER is studded with ribosomes on its body, while smooth ER does not have ribosomes on it. So that explains why the term rough and smooth used in their names. RER's main function is to transport the proteins synthesized by the ribosomes attached to it, fold them to take their final three-dimensional shape, and may also sometimes add carbohydrates to it, forming glycoproteins until forming a functional protein. SER, on the other hand, helps in lipid production, such as in testes to produce testosterone, a steroid hormone. SER is also responsible in detoxification of drugs. So, if you are a person who consumes alcohol on a regular basis, then you probably would have more SER in your liver to detoxify the alcohol you consume. Next, we have what looks like a folded piece of cloth, which is the Golgi apparatus. It is usually located near with the ER, where the cis site faces both RER and SER, while the trans site faces the cytoplasm. So, Golgi apparatus or GA's main function is to receive protein or lipid-filled vesicles from the RER and SER respectively at its cis phase, altering and modifying the proteins and lipids by adding lipids and carbohydrates to them, and then packaging them again into transport vesicles and transport them to various cells at the trans site of the GA. The vesicles can be sent to various sites based on their molecular address label, just like in the post office. Moving on to the next organelle, we have the lysosomes. It is a membrane-bound vesicle filled with hydrolytic enzymes. They are much like our stomach, where they digest materials taken into the cell, but most of these materials are unwanted by the cells, like dead organelles and bacteria. They simply have to fuse with the materials, release the hydrolytic enzymes, and destroy the unwanted particles. Now that we know about endomembrane system and the organelles, let's look at how protein production is linked with the endomembrane system. The story of protein production starts at the nucleus, where the information in the gene is transcribed into mRNA. The raw mRNA strand undergoes a process called splicing to remove unwanted parts of it. The mRNA leaves the nucleus via the nuclear pore and is used by the ribosome to form sequences of polypeptide chains. The synthesized amino acid chain enters the rough endoplasmic reticulum or RER to be folded into a three-dimensional shape of functional protein. Then, the protein is packaged in the transport vesicles to be transported to the Golgi apparatus or GA. At the GA, as mentioned earlier, protein is modified, sorted and repackaged again in secretory vesicles to be secreted out of the cell. Lastly, these secretory vesicles fuse with the plasma membrane and exit the cell to its destination. Remember that this is just a simplified version of the whole story for your simple understanding. And do forgive me for the overlap text, those were some technical problems. Hope you still could listen what I said. And with that being said, this will be the end of this video about part 1 of eukaryotic cell. Thank you everyone for watching, and I really hope you learned something new today. If you like this content and want more of it, do like this video, share it with your friends and family, and subscribe to our channel. Don't forget to hit the bell icon as well. If you have any doubts regarding any subjects, do comment your question in the comment section and we will help you to answer it. Thank you everyone and take care. Goodbye.